Welcome to the Deep Dive, where we take a stack of specialized sources, articles, and interviews and really pull out the crucial insights you need to understand the big picture.
And today, we're looking at a really fascinating paradox. It's this intersection of, well, extreme scientific achievement and deeply held religious faith.
That's right. Our main source today is an extensive interview with a truly accomplished scientist.
Oh, absolutely. He's an organic chemist, a nanotechnology expert, a professor at Rice University. This isn't just an academic. He's founded companies in pharmaceuticals, advanced electronics, even AI computing.
Okay, so let's just frame this. We have a scientist who is, operating at the absolute cutting edge. He designs molecules and materials that didn't even exist a decade ago.
He's pushing the boundaries of material science.
And yet he's also a devout Christian. He argues that his deep dive into the molecular mechanics of the universe doesn't conflict with his belief in a creator.
In fact, it's the opposite. He says his scientific understanding is what provides profound evidence for design.
And that is the core of our deep dive today. Our mission is to really explore his reasoning. Why does a scientist who spends his life at the nanoscale see this overwhelming evidence for design?
And, maybe more controversially, why does he claim that the mainstream scientific community isn't being honest about massive, huge knowledge gaps, especially when you get to the origin of life and evolution?
Let's start with that first part, the scientific awe he describes. He has this thesis that the deeper you look into how the universe is built, the more complex and, well, intentional it all seems.
Right, and he provides some really specific chemical examples to illustrate what he calls God's construction.
What are they?
He often starts with things we see every day, objects whose complexity we just completely ignore. Take, for instance, a wooden table.
Wood. Okay, we think of wood as simple, but you mentioned in the sources he sees it as this miracle of material science.
Exactly. Its impact strength is just amazing, and that's because of its precise molecular architecture. It's made of these things called polysaccharide strands.
Which are basically long chains of sugar molecules.
Right, and they're all held together by these very specific hydrogen bonds. And those bonds let the material give a little bit when it's hit.
So it absorbs energy.
It absorbs a massive amount of energy without shattering. It's this perfect, non-brittle composite.
So it's dynamically strong, not just statically strong.
Precisely. And then he says, okay, compare that to our modern materials. He points out that plastic, which comes from petroleum, it starts to decompose and fail in just a decade.
But you can find wood structures that are 1,000 years old.
1,000 years old, and the material is still structurally sound. For an expert in material science, designing something that strong, that durable, and that responsive is an engineering feat that is, frankly, beyond our current abilities.
And this is where it gets really interesting, because he then moves from a durable material like wood to, well, the fundamental process of life itself. He talks about the green leaf.
Photosynthesis.
Yeah.
It really makes you appreciate the chemistry. So why is a leaf green?
Because of chlorophyll.
Right. But chlorophyll is a light-gathering machine, and at its core, it has a single magnesium atom sitting perfectly in the middle of a complex organic cage. It's called a porphyrin ring.
Okay, a porphyrin ring. Can you just define that for us quickly?
Think of it as a highly sophisticated molecular cradle. It's this organic structure built specifically to hold that one magnesium atom in the perfect position.
So what happens?
When a photon of light, which has traveled 93 million miles from the sun, gets funnels into that ring and hits that magnesium atom, it ejects one single electron. And that starts everything.
And the result of that one perfectly managed molecular event is the entire food chain and the air we breathe.
Yes. That process takes the CO2 we exhale, uses the carbon to build the tree's structure, and then releases the oxygen. He says that this kind of knowledge, this nanoscale understanding, is what drives his awe. You look at that machine and you just have to ask, how do you pull off that level of engineering?
He even brings it back to our own bodies, right? How we process light into memory.
Oh, the process of vision is even faster. I mean, it's unbelievably intricate. When a photon hits your eye, these special molecules called rhodopsins change their shape.
And how fast does that happen?
In femtoseconds.
Yeah.
That's a quadrillionth of a second. This incredibly fast chemical reaction sends an electronic signal to your brain.
But the truly miraculous part is how that fleeting signal becomes, a permanent fact in our minds.
Right. That electronic interaction triggers protein synthesis. And this is where sleep comes in. While you're sleeping, your brain is physically converting those electrical signals into hardwired interconnects.
So you are literally building memory.
You're turning transient electrical activity into a physical permanent structure. It's amazing.
Wow. So if we connect all this back to his field, nanotechnology, it starts to make sense. The complexity is why we build our robots out of silicon and wires, not biological goo.
It's a profound point. We struggle to engineer materials that last a decade. Imagine trying to build a robot not out of silicon and copper, but out of lipids, nucleic acids, and polypeptides. Biological molecules that are dynamic, self-repairing. It's just so much more complex.
And it seems like that sheer, irreducible complexity is the foundation for his skepticism, which I think brings us to our second section, the credibility crisis in science.
Right. This is where it gets uncomfortable. The professor argues that the scientific community, particularly in these fields touching on origins and evolution, often operates under a kind of orthodoxy.
An orthodoxy that discourages honesty about what's not known.
Exactly. He says that privately, in his entire career, he's never had a single top scientist tell him, oh yeah, we have a pretty good grasp on how life started.
So in private, they agree with him.
Many do, or at least they share his deep skepticism, but they will not say it publicly.
So what's driving this? It sounds like more than just, professional disagreement.
It's systemic. It's career survival. First, there's just the human desire to be part of the crowd, to toe the party line. But really, it's about grants. It's about funding.
If you challenge the established theory, you risk your funding.
He says that's a huge part of it. And he talks about peer review bias. It is extremely easy for a reviewer, someone who has built their whole career on a certain theory, to just nix a paper that challenges their life's work. The system isn't set up for revolutions. It's set up for small, incremental steps.
And your sources detail that these aren't just hypotheticals. He faced actual real-world consequences for this.
Oh, yes. This is where the story gets really jarring. In the year 2000, he signed a very short statement.
What did it say?
It just suggested that random mutation and natural selection were inadequate to explain the diversity of life. And this is the key part, that further research is warranted.
Further research is warranted. That's like the most basic statement in all of science.
You would think. But according to his account, the reaction was immediate and punitive. He says representatives from 2 different federal agencies came to his office, no paper trail, and told him to stop writing proposals.
Because his funding would be what?
Nixed from above. That was the word used. It was active suppression of research just for suggesting more study was needed.
And it didn't stop with funding, did it?
No. Years later, 2005, he was blocked from consideration for the National Academy of Science. He had 2 colleagues sponsoring him. One was a Nobel Prize winner.
And what did they say?
They admitted to him that he had done twice as much work as was needed to qualify. But he was blackballed, explicitly because he signed that statement from 2000.
That's incredible. It really suggests that what's being defended is less a purely scientific theory and more of a, an ideological pillar.
Which brings us right to the core mystery, the origin of life, or abiogenesis. Science can define the characteristics of life, but not life itself.
And what are those characteristics that are so hard to achieve?
Well, there's responsiveness, growth, metabolism being made of cells, passing on traits. But the one he really focuses on is homeostasis.
Homeostasis. That's about maintaining a stable internal environment, right?
Exactly. It's the ability to maintain a constant internal state, like your body temperature or pH balance, with surgical precision, no matter what's happening outside. He points out that origin of life researchers often have to, water down these definitions to claim they've made proto-life or something.
They're moving the goalposts.
They have to, because their simple chemical systems have none of that dynamic complexity.
Okay, so let's get into the chemistry. What are the molecular problems that often get glossed over in press releases?
The problem starts with the basic building blocks. We're made from 4 classes of compounds: lipids, which are fats and membranes; polysaccharides, which are sugars; nucleotides for RNA and DNA; and polypeptides, which make proteins.
And his point is?
His point is that no one, using what he calls a truly prebiotic route, just the chemicals and energy available on early Earth, has ever successfully made the necessary polymers of all 4 classes.
And even if they could make them, there's this immediate handedness problem.
Yes, the stereochemistry issue. This is a huge one. The famous Miller-Urey experiment made some amino acids, sure, but it made a 50-50 mixture.
Like a mix of left-handed and right-handed molecules.
Exactly. Like your hands, they're mirror images. But in biology, all your amino acids have to be left-handed. All your sugars have to be right-handed. It's like trying to put a right-handed glove on your left hand. The machinery just doesn't work with a 50-50 mix.
And there's no known natural process on the early Earth that could have sorted them out.
None that we know of.
Yeah.
It's a severe problem. A wrong-handed molecule can shut a whole biological process down. We, as expert chemists, have to use incredibly complex methods to get that purity, yet we're supposed to believe it just happened by chance.
Which raises the ultimate challenge he poses. Even if you gave the world's best scientists all the right points of a cell, could they build one?
They cannot. Not even close. And what's so fascinating is that the knowledge gap is getting wider, not smaller.
How is it getting wider?
Because the more we learn, the more complex the cell becomes.
Hmm.
The target is moving away from them. He mentions new discoveries like chiral-induced spin selectivity.
Okay, that's a mouthful. Simple terms.
It means the inherent twist of biological molecules actually affects the spin of electrons moving through them, making them super efficient. We just discovered this. We keep finding new layers of engineering we didn't even know were there.
So if we can't figure out the first cell, the next question is how that one cell became everything else. This gets us to the evolution debate. Let's be clear about the terms. Micro versus macroevolution.
Right. Microevolution is just adaptation within a species. We see it all the time. Bird beaks changing, bacteria developing antibiotic resistance. That's observable.
But his issue is with macroevolution. The big body plan changes.
Yes. The kind of change that would turn an invertebrate with no spine into a vertebrate with a spine. He says that is never seen in the fossil record, in the way gradualism predicts.
Instead, you get the Cambrian explosion.
You get the Cambrian explosion. Around 540 million years ago, all these new distinct body plans just burst onto the scene abruptly, with no clear transitional fossils leading up to them.
And he argues there's a fundamental molecular reason why this is so difficult.
Absolutely. To change a body plan, you have to alter what are called the regulatory genetic networks.
Yeah.
These are the master controls that are laid down very early in an embryo's development.
And if you mess with those,
If you clip even one wire in that foundational circuit, he says it's almost certainly catastrophically lethal to the organism.
so there's no known molecular mechanism to explain how these massive non-lethal changes could happen.
That's his contention. And he's not alone. He notes that major developmental biologists, people like Davidson, Wagner, Irwin, have all written about this, agreeing that these gene networks pose a huge unsolved problem for the conventional theory.
Okay, so if the origin of life and macroevolution are these profound mysteries, what other major gaps in our knowledge does he point to?
He highlights a few big ones. First, sleep. We still don't really understand its core purpose. We know its function is to consolidate memory. It's when you do that hard wiring we talked about. But why we have to go unconscious to do it is still a mystery.
And the second one is on a much bigger scale.
Much bigger. We're talking cosmic gaps. Between 70 and 90% of all the matter and energy in the universe is dark matter and dark energy.
And we only know it's there by difference.
Exactly. It's missing from our calculations of the Big Bang. We have no tools to detect it directly. I mean, we are fundamentally blind to most of the universe. It's staggering.
And his final point brings it all back to chemistry, the argument from fine-tuning.
Yes. The idea that the physical constants of the universe seem perfectly set for life to exist. He uses water as an example. H2O has something called a dipole moment.
Which is about how electrons are distributed.
Right. And if that dipole moment changed by just a tiny, tiny fraction, the chemistry of water would be completely different, and life as we know it would be impossible. To a chemist, that level of precision strongly implies design, not just an accident.
So when you put it all together, the sources reveal this really strange conflict. You have a top-tier scientist presenting what seems to be rational, evidence-based skepticism.
And yet he claims he faced this fierce opposition, loss of funding, being blocked from the National Academy of Science. It's a powerful reaction.
And that reaction suggests that what's being defended isn't just a scientific theory.
It seems to be more than that. It's almost like an idea being used as an ideological weapon against the possibility of a creator. There's a great quote from the philosopher G.K. Chesterton. He said, when you stop believing in God, you don't stop believing in everything. You start believing in anything.
And the rigidity of the defense, despite these huge molecular obstacles, might point to that deeper non-scientific need to exclude a designer.
It's a possibility you have to consider.
So we'll leave you with the final provocative thought to consider. Is the constant acceleration of science, especially the mapping of the cell's complexity, that system-level engineering he described, leading us to a point where science and faith are no longer seen as contradictory?
But maybe as two different lenses observing the same overwhelming design, What does it really mean that the target of creating life moves further away every year precisely because our scientific knowledge is increasing?